So again, cells have lots of internal organelles, and they also, as we talked about, have a plasma membrane. And many of the proteins that are synthesized in cells have to wind up either outside the cell or in one of these various compartments. And so today's lecture focuses on protein trafficking, which is how proteins are targeted for different cellular components, how they get there, how they get inside. So this is a pretty big area, and we can't touch on it in much detail, but I'm going to touch on the mechanisms that I think are most useful to know about. In particular, I'll talk, these represent two of the sort of three mechanisms that are mentioned in the chapter for how proteins get inside a new compartment or inside the cell. One mechanism is by the formation of vesicles, which are just membrane-bound blobs that contain both membrane and protein. And then a specialized mechanism that enables proteins to traffic into and out of the nucleus, which is the nuclear pore, which I've touched on in a previous lecture. So this is a table I don't want you to memorize. I'm not big on memorization, but of course, if you understand the concepts, then it eliminates the need to sort of memorize terms, or the terms become sort of more familiar and less just a burden to memorize, but hopefully a way to compartmentalize your own knowledge. This table lists the major compartments that are present in eukaryotic cells, some of which we've mentioned before and some of which we'll talk about today. I'm going to wind up skipping over parts of the chapter that are sort of less familiar to me, like lysosomes. If you were taking this course from Roberto Zancu, you'd probably spend a long time on lysosomes. I know very little about lysosomes, and so I feel like you don't need to know that, but if you're interested, please go ahead and read the parts of the chapter that I cover superficially in a little more detail. This is also just not to be memorized. This is just to give you a sense of what fraction of the cell is occupied by these different compartments. And this is super variable. And I mean, the idea that the nucleus occupies 6% of the cell is maybe an average, but in some cells, the nuclei is most of the cell volume, and some it's relatively small, and so forth. Some compartments are present in just a single copy per cell, and some are present in many copies per cell. For the test, should we not focus on material from the assigned books chapters if it is not discussed in lecture? If it's not mentioned in lecture, I will not test you on it, but that doesn't mean it's not core knowledge that you should have potentially for the MCAT or for future life. But yes, for the purposes of the course, I will only test you on things that I mentioned in the lecture. Of course, the book may go into more detail than I'm able to do. Okay, so this is just a schematic of some of the major compartments in the cell. And in particular, it highlights the nucleus and the pore, which I'll talk about how things go in and out of that. And it also highlights the endoplasmic reticulum, which, as I've mentioned before, is contiguous with the outer nuclear membrane, and the Golgi apparatus. These are not to scale, really. The endoplasmic reticulum is huge. It's the major membrane-containing organelle in the cell, and it fills a lot of the volume of the cytoplasm. The Golgi is not bigger than the endoplasmic reticulum, but it is a major sorting point for proteins that are going to be targeted to various compartments, to the plasma membrane, and so it's considered to be the major sorting center in the cell. The endoplasmic reticulum is the major site of synthesis of proteins that are going to be sorted or exported from the cell. And so I'll talk a little bit more about that. So I'm going to focus on really two of the three mechanisms that are mentioned in the chapter by which proteins go into membrane-enclosed organelles or out of the cell. I'm going to talk about nuclear pore transport, and I'm going to talk about vesicle transport, and there's one slide I'll show you that just shows transport across membranes, which is how proteins get into mitochondria and chloroplasts. Okay, so at the heart of protein trafficking is this sort of code of signal sequences that are present within many proteins that are going to go somewhere within the cell. So when a protein is made, it's made in the cytosol. That's where the ribosomes are that do translation, protein synthesis. And if a protein has no signal sequence on it, then by default, it just stays in the cytosol. But if it's going to go somewhere, that information is encoded within the protein sequence by one or more peptides that direct the cell to put it somewhere special. And please do not memorize these, but it is useful to recognize that some of these sequences are present at specific points on the protein. So this import into endoplasmic reticulum signal is at the N-terminus of a protein. Similarly, mitochondrial import is an N-terminal signal. Retention of proteins in the lumen of the endoplasmic reticulum is this C-terminal sequence, KDEL, KDEL. And then other signals like nuclear import and export can be present in different locations in the protein. And you can study these signals by putting them onto another protein like GFP and watching where that goes in the cell. So these signals are both necessary and sufficient to target proteins to specific cellular compartments. And these are just examples. There are a variety of different peptide sequences that can target, for example, proteins into the nucleus. They tend to have basic amino acids, lysine and arginine, but it's not always this exact sequence. They vary. And sometimes they're what's called bipartite. They're present as two separate little peptide sequences which work together to target proteins to a location. All right, so like I said, don't memorize these sequences. I don't know them by heart, but it's useful to just see them and understand what sort of defines them in terms of where they are in the protein and their general makeup. Okay, so we'll start with nuclear pores since I've mentioned those before. Again, the nucleus is this unique organelle. It has this bilayer, this double membrane around it, and penetrating both the inner and outer nuclear membrane are these very large ring structures called nuclear pores, which are comprised of about 30 to 60 proteins that are arranged in this eightfold symmetrical organization. So they make this ring, and in the middle there's this goo, and the goo is this kind of gel-like or some kind of slimy material that gates the passage of large molecules, sorry about that, in and out of the nucleus. So small proteins that are below about 30 to 40 kilodaltons can freely pass through that material, but this material acts as a size gate, and anything larger than 30 to 40 kilodaltons, which is a fairly small protein, requires active transport in or out of the nucleus. And so import into the nucleus is encoded by nuclear localization signals, and export from the nucleus is encoded by nuclear export signals. Proteins can have actually both nuclear import and export, and by modifying those signal peptides, the cell can control when a protein goes in or out of the nucleus. So for example, if you have a nuclear localization signal and it gets phosphorylated, that might inactivate it, and that might trigger the export of that protein from the nucleus, and so forth. All right, so this is a little bit complicated, so hold on. And again, I'm happy to go through this in more detail in office hours. But the way that proteins, so this is just showing a protein that's going to get imported into the nucleus, and the export process is similar. In order to be imported into the nucleus, this protein, via its nuclear localization signal, has to bind to a carrier protein called a nuclear import receptor, also called an importin. And conversely, proteins that are going out bind to similar ferrying proteins that are called exportins, or nuclear export receptors. Collectively, those proteins are called carioferins. These transport proteins have the ability to pass through this nuclear pore material and carry passenger proteins with them. So how does a protein know to bind this nuclear import receptor here and to release it here? That's kind of the magic, and it's a little bit complicated. So I'm going to try to present it succinctly. There's a lot to unpack here. And if you're confused, as I said, I'm happy to talk in more detail about it in office hours. So the reason it's a little complicated is because we haven't really talked yet about G proteins. Hopefully you've been exposed to the idea of G proteins in other courses. If not, you may want to refresh your memory by just reading the book. So G proteins, this is what's called a small monomeric G protein. G proteins act like switches. They exist typically in two states. They can be bound to GTP, which is similar to ATP, except this is GTP, or they can be bound to GDP. And they are GTP hydrolyzing proteins. So the way that G proteins work is they bind to GTP, and usually slowly, very slowly, they will hydrolyze the GTP to GDP. And they do that spontaneously, but at a slow rate. What really affects whether a G protein is going to hydrolyze GTP and be bound to GDP, or is going to bind to GTP, is the activity of additional proteins that activate the G protein's hydrolysis activity, or activate the exchange of GDP for GTP. Sorry, I feel like I'm being a little confusing, so I'm going to restate that if I can. So this small GTPase, RAN, binds to GTP, and it hydrolyzes GTP to GDP. In the cytoplasm, there is this RAN gap protein. It's actually stuck to the outer surface of the nuclear pore. And so in the cytoplasm, this GTPase activating protein promotes the hydrolysis of GTP to GDP. And so in the cytoplasm, most of the RAN protein is GDP bound. However, when it's in the nucleus, RAN is acted on by this RAN-GEF, this guanine nucleotide exchange factor, which is actually bound to chromatin. And that triggers the release of GDP and the binding of GTP. And so in the nucleus, RAN GTP is high. In the cytoplasm, RAN GDP is high. And that's what gives directionality to nuclear import and export. So the way that this works is that importance, so again, proteins, this blue protein, which is going to pick up a nuclear targeted protein and carry it through the nuclear pore, this protein will bind to the nuclear protein in the absence of RAN GTP. And then when it goes into the nucleus, the presence of RAN GTP triggers it to release its cargo and let go of it. And so then that protein's in the nucleus, and then it will carry the RAN GTP out and do it again. So, and essentially, so the presence of high RAN GDP in the cytoplasm and high RAN GTP in the nucleus give directionality to this binding and release mechanism. And nuclear export receptors essentially do the opposite. They pick up their cargo in the nucleus in the presence of high RAN GTP, and they release it in the absence of high RAN GTP in the cytoplasm. So I know that's a little bit confusing, but if you can look at the figures and listen to that again, hopefully that will make sense. And again, I'm happy to talk about this in more detail. All right, this is the one slide I'm going to show on this mechanism of direct import of proteins through translocator proteins that are localized in the inner and outer membrane of the mitochondrion. So again, mitochondria and chloroplasts, like nuclei, have two membranes around them, but they're different structure. They don't have pores in them. Instead, they have translocator proteins, which bind to signal sequences that are present on mitochondrial or chloroplast targeted proteins. And what the textbook shows is that these receptor proteins bind to, sorry, I said the translocator proteins, but it's actually a separate receptor protein that binds to the signal sequence, tethers it to a protein translocator, and then there's a coupling between this external translocator and this internal translocator that enable the protein to traverse both the outer and inner membrane into the mitochondrial matrix. And that is literally all I'm going to say about that. And that is literally all I know about that. So if you're interested, you'll have to do some reading about more details of this process. All right, so really the rest of the lecture, I'm going to talk about vesicle transport, which is probably the most general mechanism of protein transport in the cell. And so there was a question, does every protein that is transported across membranes need its own specialized import and export? So no, so that's kind of what's shown here, that the nuclear localization signal or the nuclear export signal is what connects these nuclear targeted proteins to the importans or exportins. And likewise, these signal sequences on mitochondrial proteins are what couple them to the receptor and thus to the translocation proteins. So those are general machinery for importing things into the nucleus or the mitochondrion, as long as the signal sequences are present. Okay. So yeah, so now we're going to talk about vesicle-based transport. And this is complicated, and I'm just going to give kind of a holistic, simplified view of it. But many proteins traffic into cells, out of cells, and between different compartments in cells within vesicles, which are small membrane blobs that carry, as I said, protein and membrane with them. So most proteins that are going to be inserted into any cellular membrane or exported from the cell or targeted to a variety of different organelles in the cell are synthesized by ribosomes that are associated with the endoplasmic reticulum. And again, this is this very large, convoluted membranous structure within cells that is in some specialized secretory cells, there's just a ton of it. Some cells that make less, that traffic less protein may not have nearly as extensive endoplasmic reticulum, but this shows that probably a secretory cell that has extremely extensive endoplasmic reticulum. And this is the cytoplasm, because you can see the ribosomes in here. And then the ribosome-free areas are the lumen, the interior of the endoplasmic reticulum. And so the ribosomes associate with the membrane that is the endoplasmic reticulum. And so you've probably heard the term rough endoplasmic reticulum. That's what this is. It's this endoplasmic reticulum that's studded with ribosomes, so it has this rough appearance. So proteins that are going to enter this trafficking pathway, either a secretory pathway or that are going to wind up in the ER as their final destination, are synthesized by ribosomes on the surface of the endoplasmic reticulum. And it's the proteins that are being synthesized that actually tether the ribosomes to the endoplasmic reticulum. And this shows schematically how that works. So RNA, this is the messenger RNA that has been synthesized in the nucleus, exported into the cytoplasm, and is now going to be translated to make protein. And the way that works is that the messenger RNA is recognized by the ribosomal subunits that bind to it, and they start synthesizing protein. That's what this little green thing is. It's coming out of the ribosome. And as the ribosome moves along the RNA, additional ribosomes can bind upstream and start synthesizing. And that creates these chains of ribosomes attached to a single messenger RNA that are called, in this figure, they're called polyribosomes. Sometimes they're just called polysomes. But if these ribosomes are translating an RNA that is going to wind up making an endoplasmic reticulum protein, that protein is going to have a signal sequence at its N-terminus that is going to be recognized by a receptor on the surface of the endoplasmic reticulum, and is going to thereby tether the protein, the whole polyribosome to the endoplasmic reticulum. And this shows this very strange looking topology where the signal sequence stays embedded within this blue receptor protein, and the protein is sort of threaded into the endoplasmic reticulum, and the signal sequence is then cleaved off. So this figure doesn't show the signal recognition particle and SRP receptor. I think that's in the next slide. So this is a little bit more detail about the actual tethering of this nascent or growing polypeptide chain to the surface of the endoplasmic reticulum. So here's the signal sequence at the N-terminus of the protein. So it's the first part of the protein to be made. that signal sequence binds to this very flexible recognition protein complex called the signal recognition particle. And then that SRP docks onto a protein complex called the SRP receptor, which is embedded in the membrane of the endoplasmic reticulum. And that triggers the transfer of the polypeptide from the SRP to the SRP into this protein translocator channel. And that's what starts this threading process. And then the SRP is recycled back into the cytoplasm to go find additional proteins that have the ER signal on them. All right. And this just shows the final step in that I mentioned, which is the cleavage of the ER signal sequence by another ER membrane bound protein, which is the signal peptidase protein. So that gets cleaved off and degraded, and the protein, which no longer has this sequence on it because it's been cleaved off, enters the lumen of the endoplasmic reticulum. All right, so that didn't actually show vesicle budding. That's still coming up. This part, I had totally forgotten about this until I refreshed my memory by reading this chapter. I don't think you need to know this, but if you are very interested in membrane proteins, this and an associated video that is available with the chapter shows how more complex proteins that have transmembrane segments, which we talked about in the last lecture, get threaded into the membrane of the endoplasmic reticulum. This just shows this a single pass transmembrane protein, but a more complex process, a stepwise process, it occurs to thread multi-pass transmembrane proteins so that they wind up with their transmembrane segments embedded in this membrane. And then if they're actually supposed to be somewhere else, like at the plasma membrane, they will get trafficked out of the endoplasmic reticulum to the Golgi and then to the cell surface in vesicles, as I will show you. All right, so there was a question. Do all proteins translated on the ER have that signal sequence? Yes. Does that mean all proteins translated on an ER ribosome are going to stay in the ER? No, because they may have additional sequences that target them somewhere else. Okay, so this is what I was talking about. This shows a multi-pass transmembrane protein, but this can be more complex. Some proteins have 7 or 9 transmembrane domains, and so each one gets inserted sort of independently, and there are start and stop signals along the protein that direct the topology of the protein within the membrane. Okay, and this is something I'm not going to talk about in any detail, but it's super interesting that the ER is where a lot of protein synthesis happens, and the size of the endoplasmic reticulum is regulated by a complex system called the unfolded protein response, or UPR. And I don't keep much of this knowledge in my everyday head, although I do know that this exists. But basically, when the capacity of the endoplasmic reticulum, which, sorry, they flipped it around here, so now the lumen is shown up and the cytosol is shown down. When proteins are unable to fold properly because of a shortage of chaperones or other proteins that are important for folding up proteins and trafficking them, that actually results in a signal that causes an expansion of the size of the endoplasmic reticulum so that the cell can adjust the size and that basically the amount of endoplasmic reticulum in response to its need to secrete proteins. So if the protein synthesis needs of the cell go up, the endoplasmic reticulum will expand to accommodate that. Okay, so now I'm going to move on to how vesicle trafficking works. And if you had Bio 1A, especially if you had Randy Sheckman, you probably already learned this. So hopefully this is not all completely new to you, but if it is, then I strongly encourage you to read the book for a little bit more detail. But basically, the idea is that many transport processes within the cell occur by a process of budding and fusion. This is exocytosis. So this is a vesicle that is shown as already formed, and it's being targeted to the surface of the cell and fusing with the surface of the cell and releasing its cargo at the exterior of the cell. And then this is sort of the inverse process of endocytosis, where a vesicle is forming at the surface of the cell and carrying membrane and protein into the cell. And so this is happening all the time. Cells are constantly exo and endocytosing components from the surrounding medium, and they're also transferring material between different compartments in the cell. All of this requires a lot of energy. As we talked about before, the membranes like to be contiguous, and you can imagine that changing membrane topology by pinching off a vesicle like this and creating a new surface here require energy, and that comes from ATP and GTP and a variety of proteins that remodel membranes and pinch them off to make vesicles and fuse them to each other. All right, so I'm going to play this movie, which I think, and another one that I think help to explain and visualize these processes in a little bit more detail. This first one shows the process of receptor-mediated endocytosis, which is the process by which molecules at the exterior of the cell that bind to cellular receptors in the plasma membrane get taken into the cell.
Cholesterol circulates in the bloodstream and then enters cells by receptor-mediated endocytosis. Instead of circulating freely, cholesterol molecules are derivatized and packed inside low-density lipoprotein particles, or LDLs. A protein and phospholipid layer surrounds the cholesterol molecules. The protein portion is recognized by LDL receptors on the surface of cells. An adapter molecule, called adaptin, binds to the tail of the LDL receptor that protrudes into the cytosol. Adaptin recruits clathrin molecules, which start coating the membrane. Assembly of the clathrin coat causes the membrane to bend and invaginate, forming a vesicle that buds off inside the cell, taking with it LDL receptors and the LDL particles bound to them. Once inside the cell, the vesicle uncoats and fuses with the endosome, the intracellular compartment that first receives all endocytosed material. The endosome has low internal pH, which causes the LDL receptors to release their cargo. Empty LDL receptors are recycled to the plasma membrane in vesicles that bud off from the endosome. Each LDL receptor makes a round trip from the plasma membrane to the endosome and back every 10 minutes. Meanwhile, the LDL particles need to be disassembled. The endosomal content is delivered to a lysosome, which contains hydrolytic enzymes that can digest the particles. Free cholesterol is liberated, together with amino acids and small peptides generated by digestion of LDL proteins. The cholesterol is then released into the cytosol to be used in the synthesis of new membranes.
Okay, so that was a lot to unpack, and that was just an example of LDL being endocytosed, but that's a very general mechanism by which molecules in the extracellular fluid can be internalized within the cell. And this is a series of electron micrographs, and I'll show you a video that sort of morphs them into a movie. But what this is showing is the formation of what's called a coated pit on the surface of a cell. And all this material here is all the receptors on the surface of the cell that have been concentrated into that pit by associating with clathrin, with adapter molecules called adaptins, which then bind to clathrin on the internal surface of the membrane. And that clathrin acts to concentrate the receptors. And it also, as was just said in the video, it reshapes the membrane, it bends it, and promotes the formation of ultimately a closed vesicle coated with clathrin. So I'm going to play this video because I think this is like hard to understand, but really easy to understand if you see it in action.
Eukaryotic cells take in extracellular molecules through a process called endocytosis, in which the plasma membrane invaginates and pinches off cargo-filled vesicles. This movie shows a series of electron micrographs that have been artificially morphed to show the process of endocytosis as it may occur. The process involves a variety of molecules, including the cargo molecules that the cell takes in, the receptors that capture the cargo molecules, and molecules called adaptins that mediate contact between the receptors and the clathrin molecules that act to shape the vesicle forming at the plasma membrane. In the electron microscope, individual clathrin molecules can be seen as three-legged structures called triskelions. Each triskelion contains 3 heavy chains and 3 light chains. When a clathrin coat forms on a membrane, the globular domains that make up the tips of the heavy chains bind to adaptins, which interact with cargo membrane proteins. The assembly of a clathrin coat can occur spontaneously as numerous individual triskelions come together, interact through their leg domains, and ultimately form a closed cage.
So there was a question about whether the video links can be put into the chapter, into the slides. And as Kirsten pointed out, these videos are actually provided with your ebook, so you can see them directly in the supplemental material. And I've just picked and chosen some ones that I think are particularly helpful. If in a few cases, I've also taken videos from YouTube and other sources. And if you watch the Zoom lecture, you'll be able to see them. And I also do try to provide the links. And if you notice that I haven't, let me know and I can add them. So this just highlights that this clathrin molecule, which was featured in the video, forms this really cool structure called a triskelion, and which is made of 2 different proteins, a heavy chain and a light chain, that are each present in 3 copies to form this threefold symmetric structure. And this this structure enables the clathrin to surround the forming bud and help it to pinch off to form a vesicle. The size of the clathrin-coated vesicles varies a bit. The smallest size is about 25 nanometers, and that's just because the membrane can only bend so much and still be functional. Let's see. Yeah, what Kirsten said in the chat is accurate, I think. You can probably find these videos on YouTube too. If you just Google, you know, clathrin, you'll probably find them, but or similar ones that may be also helpful. So this just highlights a few things that were said in the video and that I've already mentioned. Again, they keep flipping around the orientation. In this case, the exterior of the cell is here and the interior of the cell is here. So molecules that are going to be endocytosed through receptor-mediated endocytosis bind to specific receptors on the cell surface. Those receptors interact with these light green adaptin proteins, which connect them to clathrin. And then clathrin, as we've already talked about, helps to bend the membrane and ultimately helps it to pinch off with the help of a GTPase called dynamin, which helps to do the final step in the fission process that separates the vesicle from the membrane that it's emerging from. And then the clathrin uncoats the vesicle inside the cell and leaves it to go to its target. Okay. So one of the things that can enter cells through receptor-mediated endocytosis is viruses. And this is a figure from the chapter. I just threw it in because I think maybe for the later I'm going to give a lecture on the cell biology of infectious disease, which is not something I know a great deal about. But again, the infectious disease that is sort of on everyone's mind is coronavirus or SARS-CoV-2. And people have been very actively researching the mechanism by which it enters cells, because of course that could be a major target for drug therapy. If we can block that entry process, then we can potentially protect people from this virus. And the exact mechanism by which these coronaviruses enter cells is still a little bit unclear. However, the protein that they use as a receptor on the cell surface is this protein called ACE2, which is angiotensin converting enzyme 2. It's not really supposed to be a viral receptor. It's actually there as part of a system that regulates blood pressure. But the virus can bind to this ACE2 protein and can use it as a way to target itself into the cell. At this point, I think it's still unclear whether it goes in through clathrin-mediated endocytosis or a clathrin-independent process. I've seen papers arguing both. So, like I said, if we can, figure this out, and if any of these proteins are potential targets for drugs, they could be a way to block the coronavirus from entering cells. ACE2, people have been looking at a lot because its expression may correlate with the vulnerability of specific patients to coronavirus infection. And for example, it's been shown that smoking may upregulate the population of cells in the airways, in the airway epithelia that express ACE2. So it may actually make people more vulnerable to infection. Okay, so I've talked about clathrin. Again, if you've ever had Randy Sheckman as an instructor, you've probably learned about other coat proteins, COP1 and COP2, which coat other types of vesicles. The video showed budding of vesicles from the plasma membrane and targeting them to endosomes. Clathrin also coats vesicles that emerge from the Golgi apparatus and are targeted to the lysosome. But vesicles that bud from the ER and are targeted to the Golgi, or vice versa, that go from the Golgi to the ER, that's called retrograde transport, are coated by a different set of proteins called COP1 and COP2. And that is all I'm going to say about those, and literally all I know about them. So again, feel free to read further, but that's all I expect you to know. All right, so we've talked about the targets on proteins that target them to specific parts of the cell, or these various signal sequences. But once they're incorporated into a vesicle, the vesicle also has to be targeted to the right place in the cell, and that is through an additional sort of matchmaking system within cells formed by snare proteins. So these vesicles have surface proteins that are, again, small GTPases, and those actually serve as a code for docking on specific compartments within the cell through tethering proteins. So this is the recognition system. The tethering protein recognizes a specific Rab GTPase on the surface of a specific class of vesicles. And then these T snares and V snares, so target snares and vesicle snares, form pairwise interactions that also add some specificity to this docking process. And they facilitate the close apposition of vesicles to membranes that they're going to fuse with. And then there are additional proteins that carry out this step of actually fusing the vesicle to the target membrane, which again is an energy intensive process. It's very energetically unfavorable to bring this membrane in close proximity to this other membrane, because again, there are these hydrophilic head groups on the membrane, and you've got to squeeze out the water and ultimately allow these membranes to fuse with each other, and that requires energy. Okay, so both Rab and B snare confer specificity, or just one or the other? My understanding, which is not very deep, is that both confer specificity. That I know that the Rabs do for sure. There's many different Rab proteins, and so they form a code that is recognized by these tethering proteins. And I think that there are also different B snares and T snares that add specificity to this process, but I will check that out, and we can talk about it at office hours if you want to go into more detail. Okay, so, and this just shows again the role of the pairwise interactions between the T snares and V snares that help to mediate this energy intensive fusion reaction. Okay, so I'm going to talk a little bit about the Golgi. I always found this a very confusing organelle. It looks so weird. The book illustrates it as a stack of pita bread, which is, I think, somewhat helpful in understanding the topology of this organelle. So the Golgi is it's a whole stack of different compartments that are called cisternae, which literally just means compartment. And if you took Latin or some Romance language, it may help you in thinking about what these different compartments are. But you may also remember from organic chemistry that cis means nearby and trans means far away. So in this case, nearby is the endoplasmic reticulum. So I talked about how proteins are synthesized at the endoplasmic reticulum. And then if they're not going to stay in the endoplasmic reticulum, chances are their next stop is the Golgi networks. They bud out of the endoplasmic reticulum into a vesicle, and that vesicle then buds to the cis Golgi network and becomes part of the cis Golgi network. And so it's the budding of vesicles that form these cisternae and cause them to grow. And then there are also vesicles constantly budding from each of these cisternae and moving to, well, it's not so much that they that the vesicles carry things from the cis to the medial to the trans. That's not actually how it works. What actually happens is that these compartments mature by having vesicles carry things away from them that are specific to the cis network and going to other compartments. And then other vesicles join these compartments and bring in components that make them part of the medial cisternae, and eventually the trans cisternae. I have a nice video that I'll provide a link to that I think helps to illustrate that better than I just explained it. But basically, these compartments undergo maturation from cis to medial to trans by having things come into them and leave them through these vesicle transport processes. And a lot of what's coming in and going away are modifying proteins that glycosylate, that add sugars to the proteins within these compartments. And by doing that, they add another layer of direction to the proteins within these compartments that will ultimately contribute to their targeting to the extracellular space or the plasma membrane or different compartments within the cell.
It is important that translated proteins are delivered to their specific cellular location. To accomplish this, the protein is transferred through a series of membrane structures. A principal membrane component is the Golgi apparatus. The Golgi apparatus is the sorting organelle of the cell. Proteins from the rough endoplasmic reticulum are sent to the Golgi. As the proteins move through the Golgi apparatus, they are modified and packaged into vesicles. Because the Golgi apparatus receives proteins from one location and targets them for delivery to a second location, it is sometimes considered the post office of the cell. The Golgi apparatus consists of general components: the cis cisterna nearest the endoplasmic reticulum, or ER, the medial and trans cisternae, and the trans Golgi network. Other key players in this process are the proteins being transported and the enzymes that modify them. Translated proteins are encapsulated in vesicles in the ER. A group of these vesicles fuse, and these fused vesicles form the cis cisterna. As the protein moves through the stack, it is modified by resident Golgi enzymes at specific locations in the apparatus. These modifications are important because they provide the signal that determines the final destination of the protein. So how does the protein move through the Golgi? Movement occurs in waves. First, the cis cisterna becomes part of the medial Golgi cisternae. Behind it, a new cis cisterna is formed by the fusion of vesicles from the ER. Meanwhile, one of the medial cisternae migrates and becomes the new trans cisterna. Collectively, this process is known as the cis maturation model. Proteins are sorted within the trans Golgi network. Proteins with the same target sequence are destined for delivery to the same location. The trans-Golgi network then buds off into vesicles. These vesicles then migrate to their target location. These locations include internal organelles, such as the lysosome, the digestive organelle of the cell. The vesicles can also be targeted to the cell membrane, where the targeted protein can be released from the cell for delivery elsewhere in the organism.
Fluorescently labeled membrane proteins start their journey to the plasma membrane after synthesis in the endoplasmic reticulum. They are first dispersed throughout the extensive membrane network of the endoplasmic reticulum, from where they move to exit sites that form in random locations in the membrane network. At each of these sites, the membrane proteins are concentrated and packaged into transport vesicles. Clusters of the transport vesicles fuse to form transport intermediates. At the next stage, transport intermediates move along microtubule tracks to the Golgi apparatus near the center of the cell. The membrane proteins exit the Golgi apparatus. They move in transport vesicles that are now pulled outward on microtubules, which deliver them to the plasma membrane. Each time a Golgi-derived vesicle fuses with the plasma membrane, its content proteins disperse.
Well, the secretory membrane system is a system characteristic of all eukaryotic cells. It is all of the membranes within this system are bilayered, and they are responsible fundamentally for allowing cells to secrete various constituents. This system is also responsible for the formation of the nuclear envelope. And prior to GFP technology, our understanding of the geography and the dynamics of this system was quite limited, relying primarily on static images, really just snapshots at different time points. So there was many questions that were very unclear. But what I want to take you through is some of the data that has really provided a new type of foundation for our thinking about these organelles. Starting with the endoplasmic reticulum, what we're looking at here is a GFP tag marker that targets to the endoplasmic reticulum. And you can see in vivid detail the elaborate reticular network that defines this key organelle that serves as the site of all protein synthesis that is devoted to the secretory system. In fact, all the proteins that make up the endomembranes within the cell are derived by initially being synthesized in the endoplasmic reticulum. Okay, well, what about secretion? GFP has played a huge role in allowing us to visualize the trafficking intermediates that define the secretory pathway. Now, this pathway involves the movement of molecules from the ER through a series of substations that define the secretory pathway. And a key substation is the Golgi apparatus, which sits in the juxtanuclear area of the cell. Now, if we visualize how molecules traffic through this system, one has the ability to define the steps of this pathway. And in the movie that I'm going to be showing you, what we're going to be looking at is one type of protein, one of many proteins that move through this secretory pathway. We've tagged this protein with GFP in order to be able to visualize it in this cell. The protein has a characteristic of misfolding and being retained in the ER at a restrictive temperature of 40 degrees. But upon shift to the permissive temperature, which allows folding of the VSVG, the molecule can then move through the secretory pathway. And that temperature shift can be utilized, as shown here, to synchronously release a population of these molecules from the ER to watch them pass through the secretory pathway. And that's what you're seeing right here in this movie. The molecules are moving, in fact, there's 20 million of these molecules that are moving sequentially from ER to Golgi to plasma membrane. Now, we can also look at the movement of these molecules Once they've been situated in the Golgi, where they undergo modifications, carbohydrate modifications, that's the function of the Golgi. But as you can see in this movie here, these transport intermediates are move in the opposite direction, out from the Golgi, in order to reach the plasma membrane. Now these transport intermediates are moving along microtubule tracks, just as the ER to Golgi transport intermediates are doing. And they're moving out to sites on the plasma membrane where they can undergo a fusion reaction. And again, they're moving along microtubules as well, but in the opposite direction along microtubules.
Microtubules form this astral array within the cells, like a radial array that is nucleated and centered at this organelle called the microtubule organizing center, which we will talk about in a few lectures. And the Golgi is basically associated with the microtubule organizing center. So I'll have more to say about that. And there was a question about the textbook. I had posted a link about how to access the first 2 chapters in the textbook, and then I deleted that and posted a way that you can actually access the entire textbook. So hopefully most of you have done that already. All right.
Someone also wanted clarification. Sorry. Someone asked that, basically asked for clarification about cis and trans.
Sorry.
I'm not sure.
Okay, so cis means close and trans means kind of away. And as you just saw in that video, secreted proteins and many organelle targeted proteins are synthesized at the endoplasmic reticulum, again, the rough endoplasmic reticulum. And once they're made, then their next stop is the Golgi. And so they go to the Golgi and they enter the Golgi. And so initially that's called the cis Golgi. And then as these proteins are processed in the Golgi, the compartments that they're in within the Golgi are undergoing maturation. And they do that by having vesicles carry stuff away that they no longer need, and by having additional vesicles fused with them that carry new processing enzymes. And so the Golgi compartments undergo this maturation from cis, which means close to the ER, to trans, which means far from the ER. And from the trans Golgi, that's where things butt off and go to other destinations in the cell.
Passenger proteins exiting the Golgi apparatus on the way to the cell surface are often packaged into tubular transport vesicles of significant size. Such tubular vesicles can branch and fragment before they fuse with the plasma membrane. The transport vesicles move along microtubules, which are stained here with a red fluorescent dye. The green cell in the corner does not contain fluorescent microtubules.
So the main point of that video, beyond what I had already shown you, is just that as things bud off of the endoplasmic reticulum, you can see these kind of large tubular vesicles, and also that many vesicles are moving along microtubules within the cell, and they can move inward from the plasma membrane along microtubules, and they can move outward to the plasma membrane along microtubules using different motors based on the polarity of microtubules, which we have not yet talked about in any specifics. So we'll come to that later. Okay, and as I said, if you've ever taken a class from Randy Sheckman, or maybe others within our department, you have likely heard about parts of the secretory pathway before, and this is just a reminder that Randy Sheckman, our local hero who won the Nobel Prize a few years ago, was a major player in understanding how these processes that I've just described and the videos just described, how they work. And he and his lab took a genetic approach to understanding this process of secretion. They had this insight that you could isolate yeast cells, budding yeast cells that had defects in the secretory pathway, because it turns out that those cells accumulate proteins that are supposed to be exported from the cell. If they can't be exported from the cell, they pile up inside the cell and make the cells denser. And so they isolated these dense yeast cells, and then using a variety of tools, they figured out which genes were mutated in those yeast cells, and through those analyses, they were able to figure out the pathway, the molecular pathway by which all these processes of secretion happen, at least within yeast cells. And most of that is, of course, conserved with mammalian cells. Okay, and I think that was all I was going to say today. So I actually made it within the hour just, which is amazing. I'm happy to take questions now. I would really encourage you to come to office hours tomorrow if you want me to talk about this material in a different way, or if you are confused about anything. And this book, I mean, we could talk, again, this could be the subject of an entire course. This is a very sort of superficial description of different protein trafficking pathways, but hopefully it gives you a sense of how things move around within cells, how proteins encode their own localization information. And any questions now?
Oh, someone wants to ask
Will
if
office hours be recorded? Yes, I'll record office hours. If anyone shows up. Last week, nobody showed up. I just sat on my couch and felt sad. But come to office hours, and Paul will be here with me. And you can see my Paul Red pillows over there. I have a lot of Paul Red paraphernalia. Is Zoom for office hours the same as for lecture? Yes. And good to see those of you who are here, and hope you're all doing okay. See you soon.
Someone was asking if they'll receive practice problems or pass the midterms to help prepare for the final.
That would be a good thing to do. Since I have not taught this course for a decade, I haven't written questions for this before, but I will try to find relevant questions. And then I still want to try to figure out how to make this smart work thing happen, but I wasn't able to do that. We'll see if I can get that. No, someone asked about big quizzes this week. That was announced recently, that the big quiz, although it was initially on the syllabus for this week, that was before we extended the time for the first midterm and delayed the beginning of this section. So the big quiz is next week. And yes, specifics to be determined and will be announced shortly. Any other questions?